What we are doing is just adding some genetic modification of T cells to make these cells available as an off-the-shelf product to be able to treat patients on an autologous, on an allogenic fashion.
Can you tell me what it is normally that is done? Because the cures we've seen have been with autologous transfer using the so-called CAR T-cell therapy. What exactly is that and how does it work?
Now, what's called the autologous adoptive T-cell immunotherapy is clinicians will take the T-cells from the patient, will infect them with a modified virus that will bring a chimeric antigen receptor to the cell, and this receptor will recognize a tumor antigen, and in that way the cell will kill the tumor cell. So what is happening with this in this context is the cells come from a patient and they are injected back in the same patient. In fact, it's a direct killing of the cells. So the modified cell have a high specificity because it has the binding domain of a monoclonal antibody, and it will perfectly recognize the antigen, bind to the tumoral cell, and drive the killing of these cells.
And there have been some fairly dramatic cures with this therapy, but of course it's only been in a handful of patients because you have to make the chimeric antigen receptor T-cell therapy individually each time. So what is it that you're doing? How could this change?
Our approach is that we have been developing nucleases for several years. So these nucleases will recognize a specific gene, cleave it, and inactivate the gene. So we have combined this CAR T-cell technology with genome engineering to inactivate, for example, the T-cell receptor, and in that way a cell that can be issued from a healthy donor could be modified and treat in an allogenic fashion many patients. So far we have been efficiently doing this genetic modification. This multiplex genome editing means that we can have some double knockouts of different genes, and we have validated their anti-tumoral activity in mice models. Yeah, that's what we hope. We are planning to do our first in-man test in 2015, so we are working hard to get it into the clinic. I think they should be as effective as the autologous T-cell. The advantage is that in a single production run of these CAR T-cells, you can treat many different patients. For the moment, everything that has been tested is mostly on hematological cancers, and that's what we are developing, but we are now trying to find different genetic modifications that would help us to target tumor solid also. The first candidates we are working on are, for example, targeting the CD123 antigen in acute myeloid leukemia patients, and also targeting the BCMA gene in patients with multiple myeloma. The clinical potential is that you can have an off-the-shelf therapeutic cellular product that could be infused into patients rapidly. You don't need to take the patient own cells and develop all these modifications, which is a long process, an expensive process that you need to know how to do it. So in this case, the cells will be ready to be injected to the patient, and it will be much faster than these autologous approaches. Yeah, we are also working on that, so we hope to. There is more difficulty in treating the solid tumors, but we think it can be done for sure. I think that the take-home message is that we are developing this off-the-shelf platform that could be easily upscalable and easily modified to target different kinds of tumors. So it's, I think, some technology that needs to be attentive to it, because it will change the way we treat cancer in the future. It's something that many patients are asking for these kind of treatments, because all what we have seen in the news is amazing. It's revolutionizing the way that people is facing cancer and how you see that it can be done. So I think that patients should be aware of these kind of technologies and these alternative treatments. Yeah, no, it's available for patients that have some T cells in good conditions that will allow the production of CAR-expressing T cells. But hopefully, if we have an allogenic product, it will be available for many, many people. We are now starting the development of the preclinical and clinical studies. And we hope to be able to be starting our first-in-man test in 2015. So once we prove that there is efficiency of this technology, of this approach in treatment of lymphoma, we can move to the other pathologies and speed up the process of developing this kind of product.